Hello everyone, my name is Salakshi and I'm a first year MBBS student, and today I will be teaching this chapter Metabolism of Cholesterol, which is a chapter of Metabolism of Lipid, and this is one of the important topic. So let's begin with this. So Metabolism of Cholesterol. So cholesterol is main sterol in animals and it is amphipathic in nature. Matlab it is polar as well as non-polar. It contributes to both the structures, and the topics which I will be covering under the heading Metabolism of Cholesterol is synthesis of cholesterol, then functions of cholesterol, regulation of cholesterol synthesis, and degradation of cholesterol. So these are the main four subheading under the topic Metabolism of Cholesterol. So let's first begin with the synthesis of cholesterol. So next is synthesis of cholesterol, which is the first topic in Metabolism of Cholesterol. So synthesis of cholesterol major contribution is by liver and intestine, and the enzymes which are required for synthesis of cholesterol are present in cytosol. So for the synthesis of one cholesterol, 18 acetyl CoA, 36 ATP and 16 NADPH are required. So synthesis of cholesterol takes place in mainly 5 stages. So first is HMG CoA synthesis. So HMG CoA is 6 carbon compound. Then mevalonate synthesis, which is 6 carbon. Isoprenoid unit production. Isoprenoid unit is out of 5 carbon. Squalene formation, and next is and fifth is cholesterol synthesis, which so squalene से cholesterol बनता है in the last step. So these are the 5 important stages of synthesis of cholesterol. Now let's begin with all these stages in detail. Synthesis of cholesterol में पहला और the first step was HMG CoA synthesis. So HMG CoA यहाँ पे बनता है at this step over here. Okay, so this is basically our first step. So 2-acetyl CoA with the presence of the enzyme thiolase, it forms acetoacetyl CoA, and then by the enzyme HMG synthase, it forms HMG CoA. यहाँ पे it requires 1 ATP. So this is the first step in cholesterol synthesis. Now the second step, that is mevalonate formation. So this step is important as it also involves in the regulation of cholesterol synthesis. So HMG CoA, it forms mevalonate by the enzyme HMG reductase. So this is the rate limiting enzyme. यहाँ पे 2 NADPH plus H plus से बन के 2 NADP plus बनता है. So this is the reducing equivalent. Now mevalonate से the third step. This is basically the second step. Now the third step. So the third step is bigger. So हमारा third step में था mevalonate से isoprenoids बनते थे ना, units बनते थे. So they are basically IPP and DMP. So isoprenoids units. So यहाँ पे मतलब मैंने पूरा दिखाया नहीं है. So third step I'll show you. So the first is the mevalonate. So mevalonate से बनता है 5-phosphomevalonate. यहाँ पे ATP is used. So the enzyme is kinase. So it's mevalonate kinase. Then again phosphophenol mevalonate, मैंने फिर से नहीं लिखा है. So it's phosphophenol mevalonate kinase. It's mevalonate kinase. So मैंने सिर्फ kinase लिखा है, तो इतना समझ लो. So mevalonate kinase से then 5-phosphophenol mevalonate बनता है. Again ATP is common in every step. So again 5-phosphophenol mevalonate kinase, the enzyme. Then it forms 5-pyrophospophenol mevalonate. Again by the help of the enzyme kinase, that is pyrophosphomevalonate kinase. Pyrophosphomevalonate kinase, it forms 3-phospho-5-pyrophosphomevalonate. So this is third step. It's not complete, of course. I'll show you in the next part of the video. So यहाँ तक आ गए. So you can note this down and take a screenshot. Then I'll show you the next step. Let's complete the third step. So 3-phospho-5-pyrophosphomevalonate with the enzyme pyrophosphomevalonate decarboxylase. So basically remember decarboxylase क्योंकि ऊपर वाला तो ऐसे ही है, मतलब याद करने के लिए. So pyrophosphomevalonate decarboxylase, it form isopentyl pyrophosphate. and then with the enzyme isomerase it formed dimethyl allyl pyrophosphate. So ये जो दो है जिनको मैंने स्टार मार्क किया है, they are isoprenard unit, they are 5 carbon. So this is the third step. Now we'll move towards the fourth step. Okay. जो हमारा लास्ट थर्ड स्टेप में बना था आइसोप्रेनॉइड यूनिट डायमेथाइल एलाइल पायरोफॉस्फेट, उससे नाउ विल मूव टुवर्ड्स द फोर्थ स्टेप विच इज़ द फॉर्मेशन ऑफ स्क्वालिन ओवर हियर. सो ये भी थोड़ा लंबा है, सो यहाँ पे अपना फोर्थ स्टेप कंप्लीट हो रहा है. सो इट्स बाय द एंजाइम सिस्प्रेनाइल ट्रांसफरेज इट फॉर्म guanylyl pyrophosphate which is of 10 carbon, and again by same enzyme it formed faranyl pyrophosphate. Then by the enzyme squalene synthase, and यहाँ पे भी reduction equivalent NADPH plus H plus से NADP plus बनता है, squalene बनता है which is 30 carbon compound, and then by oxidase hydrolase kylase इससे अपना लैनोस्टेरॉल बनता है जस्ट 30 कार्बन, एंड देन बाय अ 19 रिएक्शन सीरीज से फाइनली कोलेस्ट्रॉल इज फॉर्म. सो दिस इज द सिंथेसिस ऑफ कोलेस्ट्रॉल. So basically you don't need to memorize it in a paragraph form, you can simply just write this in diagrammatic form. So yeah, so 5 steps से बनता है HMG CoA synthesis which is formed by acetyl CoA. So basically acetyl CoA से HMG CoA बनता है, बीच में acetyl CoA भी बनता है. So thiolase enzyme, so acetyl CoA with the thiolase enzyme it form acetyl CoA, then HMG synthase enzyme से HMG CoA बनता है. फिर जैसे जैसे स्टेप्स आगे बढ़ते हैं इट फॉर्म्स फाइनली कोलेस्ट्रॉल. सो दिस इज हाउ कोलेस्ट्रॉल इज फॉर्म. सो मेटाबॉलिज्म ऑफ कोलेस्ट्रॉल का फर्स्ट पार्ट कंप्लीट हो गया, दिस सिंथेसिस. नाउ विल मूव टुवर्ड्स द सेकंड पार्ट दैट इज फंक्शंस ऑफ कोलेस्ट्रॉल. इट्स छोटू सा टॉपिक. सेकंड सब टॉपिक इज फंक्शंस ऑफ कोलेस्ट्रॉल. So functions of cholesterol, cholesterol is structural component of cell membrane. तो सेल मेम्ब्रेन में कोलेस्ट्रॉल होता ही है, सो इट इज़ द स्ट्रक्चरल कंपोनेंट ऑफ सेल मेम्ब्रेन. देन इट इज़ द प्रिकर्सर इन सिंथेसिस ऑफ स्टेरॉइड हार्मोन, विटामिन डी एंड बाइल एसिड्स. Then it is important ingredient in the structure of liver protein, and it also helps in the transport of fatty acids to liver. So these are the 4 main important functions of cholesterol. Now after we have completed synthesis of cholesterol, functions of cholesterol, now the next is the regulation of cholesterol. Now let's move towards that topic. You can take screenshot. The regulation of synthesis of cholesterol. So हमने जो एंजाइम देखा था इन द सिंथेसिस ऑफ कोलेस्ट्रॉल के जो स्टेप्स थे उसमें हमने एक एंजाइम देखा था HMG CoA reductase. So it is the rate limiting enzyme. I've told you before also. So it is the rate limiting enzyme. So this enzyme control the regulation of synthesis of cholesterol. कौन से स्टेप में देखा था? So हमारा जो HMG CoA है इससे मेवालोनेट बनता है. So here is the enzyme which helps in the formation of this. That is HMG CoA reductase. Basically, it's the second step in the synthesis of cholesterol. So now, how does HMG CoA reductase enzyme regulate the synthesis of cholesterol? So it is basically by 4 things. एक है feedback control. Second is hormonal regulation. Third is inhibited by drugs, and fourth is bile acids. I'll tell you how this occurs stepwise. So let's begin with the regulation of synthesis of cholesterol. First is the feedback control. So इसमें क्या होता है? So we know कि यहाँ पे cholesterol synthesis हो रहा है. So increase in the concentration of cholesterol. I'm writing CHO for cholesterol. I'll give you now. Increase in the concentration of cholesterol. So this causes a negative effect on the enzyme HMG CoA reductase. अगर कॉन्सेंट्रेशन ऑफ कोलेस्ट्रॉल कोलेस्ट्रॉल बढ़ गया है, मतलब कोलेस्ट्रॉल सिंथेसिस ज़्यादा हो गया है, तो HMG CoA रिडक्टेज का जो प्रोडक्शन है वो कम हो जाएगा. सो इट इज़ बेसिकली फीडबैक कंट्रोल मैकेनिज्म. वो अब प्रोडक्शन कम कैसे होगा? सो बेसिकली इट ऑकर्स बाय डिक्रीजिंग decreasing transcription of gene that is regulating responsible for HMG CoA reductase formation. सो बेसिकली कोलेस्ट्रॉल सिंथेसिस मतलब कोलेस्ट्रॉल बढ़ गया तो HMG CoA रिडक्टेज का प्रोडक्शन कम हो जाएगा. कम कैसे होगा? सो बेसिकली इट इज़ बाय डिक्रीजिंग ट्रांसक्रिप्शन ऑफ जीन रिस्पॉन्सिबल फॉर HMG CoA रिडक्टेज फॉर्मेशन. सो दिस इज़ द फर्स्ट थिंग फीडबैक कंट्रोल जिससे हमारा सिंथेसिस ऑफ कोलेस्ट्रॉल को रेगुलेट किया जाता है. नाउ लेट्स मूव टुवर्ड्स द सेकेंड थिंग दैट इज हार्मोनल कंट्रोल. सो दिस इज हार्मोनल रेगुलेशन. सो द एंजाइम्स HMG CoA reductase, it has two interconvertible form. First is phosphorated, which is inactive, and next is dephosphorated, which is active. So the hormones glucocorticoid and glucagon. Check the spelling, I'm not sure with this. So ये जो hormones है, this increases the formation of inactive HMG CoA reductase enzyme basically. सो इसके बाद अगर इनएक्टिव HMG CoA रिडक्टेज एंजाइम बन गया सो इससे सिंथेसिस ऑफ कोलेस्ट्रॉल डिक्रीजेस. सो इन हार्मोन्स से सिंथेसिस ऑफ कोलेस्ट्रॉल कम होगा. नाउ डीफॉस्फोरेटेड मतलब जो एक्टिव HMG रिडक्टेज है सो द एंजाइम्स लाइक इंसुलिन ये इसका फॉर्मेशन बढ़ाते हैं जिसके वजह से synthesis of cholesterol increases. So basically this hormone increases the synthesis of cholesterol and these hormone decreases the मतलब synthesis of cholesterol decrease करता है. So this is how hormonal regulation of HMG CoA reductase enzyme occurs. Now let's move towards the drugs, that is in how synthesis of cholesterol synthesis of cholesterol is inhibited by drugs. So let's see that. Inhibition by drugs. So the drug compactin and levastatin is another drug which decreases the synthesis of cholesterol as they are the competitive inhibitor for the enzyme HMG CoA reductase. And the fourth regulation is that bile acid basically decreases HMG CoA activity, so it decreases synthesis of cholesterol. So these are the four things which regulate the synthesis of cholesterol. Now let's move towards the degradation of cholesterol. Last topic in the metabolism of cholesterol is the degradation of cholesterol. So cholesterol का basically bile acids, steroids, vitamin D ये सब बनता है. So यहाँ पे हम देखेंगे degradation of cholesterol. So basically I have written this in a tabular form. So you need to memorize this because it's not much to explain. So basically cholesterol का 7-hydroxy cholesterol बनता है by the enzyme 7-alpha hydroxylase. Then by several steps से it forms two things. एक तो cholic acid and next is cholic acid. Now उसका आगे का हम देखेंगे. So next is the degradation of cholesterol. मतलब जो हमने स्टेप्स देखा था हम कॉलिक एसिड और कीनोडीऑक्सीकॉलिक एसिड तक आ गए थे. So now by this tyrosine and glycine it formed tyrocolic acid and glycolic acid, which by intestinal bacteria it formed deoxycholic acid, and here by same things it forms tyro or glycodeoxycholic acid, which by intestinal bacteria it formed lithocholic acid. So this was the degradation of cholesterol. So in the metabolism of cholesterol we have covered synthesis, function, regulation and degradation of cholesterol. So these were the four main topics in the metabolism of cholesterol, which is a topic in metabolism of lipids. So yeah, I hope you have understood this. Thanks for watching the video. Do like, share and subscribe. Thank you.